Good afternoon, good morning. My name is Adam Sherman, Research Director at the IFLPA. I'd like to thank everyone for taking time out of your busy schedules to participate in today's talk. Today's webinar will cover Regeneron's LUMINA-1 clinical trial, which is a study looking at the safety and effects of Regen-2477, which is an investigational drug for FOP. Giving this talk is Dr. Bob Pignolo, Professor of Geriatric Medicine at the Mayo Clinic College of Medicine. Dr. Pignolo is a trial and site investigator for the Illumina clinical trial. Also participating, we have Dr. Scott Mellis, Vice President, Early Clinical Development at Regeneron Pharmaceuticals, who will be available during the Q&A portion for the call. Before we begin, I just had a few housekeeping items. First, you can submit your questions through the Q&A function on the webinar. This is a button which can be found at the bottom of your screen. Questions will be held until the end of the talk, at which point we'll pose your questions directly to Drs. Pignolo and Mellis. All questions will be kept anonymous. Next, we will be recording this webinar for those who are unable to attend today's session. And then finally, just to note, we have conducted prior webinars, and the recordings of these talks can be found on the ifopa.org website. With that, I'd like to turn it over to Dr. Bob Pignolo.
Thank you, Adam. Thank you very much. So the title of today's webinar is Anti-Activin A and FOP, and we'll be talking about clinical research, preclinical research, and the LUMINA-1 clinical trial. By way of relevant disclosures, my name is Bob Pignolo, and I'm the principal investigator here at Mayo Clinic for both the Clementia and Regeneron clinical trials. So this is an outline of the webinar presentation. I'll start with some clinical challenges that are known to all of you in terms of FOP. And then I'll give a preclinical FOP research update, some reflections on clinical research related to the translation of the preclinical research I'll discuss into the current study. And then I'll spend the rest of my time discussing elements of the Illumina-1 or anti-activin A clinical trial, including the goals, who can participate, study design, medication, and where to find more information. So as we're all well aware of, some of the major clinical challenges of FOP is in the setting of flare-ups with swelling, pain, tenderness, a whole constellation of signs and symptoms which eventually lead to the formation of new bone outside the normal skeleton, causing reduced motion about joints, altered growth, and often very severe pain. I should mention at this point that in almost 50% of patients with FOP, FOP continues to progress in the absence of flare-ups. And I'll talk a little bit about this later as well, but the imaging modality that we're using in the Illumina-1 trial offers the possibility to detect activity of bone formation, potentially even in the absence of flare-ups, but a little bit more on that later. So let's begin with a discussion about the preclinical studies that led to the use of the activin A antibody in the Illumina-1 trial. So Regeneron created a mouse with the FOP gene containing the classical ACVR1 R206H mutation. And this model has been shared with colleagues in academia and industry, and I think has been a very fruitful addition to advancing preclinical research in FOP. At the same time that this model was being developed, additional cellular studies pointed to the unexpected activity of a protein called activin A on the mutated receptor. And so with these tools in hand, Regeneron embarked on different lines of evidence in both basic and clinical research that have led to the current clinical trial. I'm going to present this data over the next couple of slides, but please remember that the data I'm going to show you are from mouse models in the laboratory, and it's really the reproduction of the exciting results in the laboratory really awaits the completion of ongoing clinical trials. So the first point is that the R206H FOP mouse model resembles human disease. So heterotopic bone, or bone outside the normal skeleton, forms and expands, causing impaired mobility. So you can see in both figures that the heterotopic or extraskeletal bone is colored yellow here, just so you can see where it is. And the arrows indicate the direction of the growth of the heterotopic bone. So it's not only the case that heterotopic bone is formed, but after it forms, it continues to grow and expand. And you can see, especially on the left, that because the heterotopic bone is growing in this case 3 different directions, it's ankylosing not only to the femur, but it's also ankylosing to the tibia and the tail of the mouse. And on the right, you can see how the heterotopic bone is growing down the spine. So in each case, these scenarios are very similar to what occurs in patients with FOP. So in this experiment, this is sort of a proof of concept experiment where activin A, not the antibody, which is the investigational drug in the current clinical trial, but activin A stimulates new bone in the FOP mouse model. So you can see the red arrow pointing to an area where a collagen sponge containing activin A was implanted into the thigh muscle. And you can see the activin A by itself only in the FOP mouse, not in a non-FOP or wild type mouse, does the extra bone form. So at the time that this model was developed, an antibody to activin A was available at Regeneron, and this began the preclinical journey that led to our knowledge of its efficacy, at least in a mouse model. So what you see here is a schematic of an antibody. On the left, that's a 3D representation, and then on the right, the cartoon version of the antibody, which binds activin. This is the ligand or the molecule that binds to the mutated receptor and activates it. In the presence of the antibody sequestering activin, there's inactivation and prevention of the interaction of activin A with the mutated receptor, and in doing so, signaling is decreased through the mutated receptor. So as you know, antibodies are a part of our immune system, and we normally think of them as helping fight infection. But in this case, they were created in the laboratory to be very specific and to target, in this case, a molecule called activin A that was found to increase signaling through the mutant receptor. So the laboratory at Regeneron used this model in 3 different scenarios to see if the activin A antibody could prevent heterotopic bone from forming before the process has started, if it could halt progression of heterotopic bone once the process had already started, and finally, could the antibody induce regression of some bony lesions once they were already there. So this is the first scenario. So if you give anti-activin A antibody prior to formation of heterotopic bone, indeed, anti-activin A can prevent its formation. So in the left panel, you see the situation where a non-specific antibody is given. It's called an isotype control. So this is an antibody that is not, does not have any affinity towards activin A. In the right panel, circumscribed in red, that animal did receive the antibody, and as you can see by looking at the extra bone colored in yellow, there was no bone formed on the right, only when the control antibody was given. So can anti-activin A antibody prevent the progression of heterotopic bone when given after the bone has started? And that's what this figure shows. So in the solid line, in the solid black line, that is the accumulation of heterotopic bone over time. The time is shown along the bottom in weeks. So you could see that by week 3, and certainly after week 3, in the animal given the control antibody, that there's an accumulation of total heterotopic bone. In contrast, if you look at the dashed red line, you can see that at the start of treatment, again on week 3, that not only did the anti-activin A antibody halt the progression of heterotopic bone, but we actually see a little bit of regression through week 6. And then finally, can the activin A antibody cause regression of lesions? And so here we have the same animal on day 21 and on day 42, and the anti-activin A antibody is given to that animal after the heterotopic bone is formed. And in this particular case, you can see some regression of the heterotopic bone. This does not occur in every animal, but this is nevertheless a very interesting finding that needs to be investigated further. So just to summarize the preclinical data, anti-activin A antibody can prevent heterotopic bone when given before the extraskeletal bone starts forming. It can halt progression of heterotopic bone after the heterotopic bone has started, and in some cases it can even mediate regression of new heterotopic bone. Okay, so just some thoughts on clinical research and how one goes about translating preclinical data into people. So of course, these are the important considerations: safety first, a rigorous design to provide adequate evidence to decide if something has if an investigational drug has a meaningful effect or effects, good clinical practice and study oversight. So obviously getting studies approved by institutional review bodies and regulatory authorities. And then timely and transparent data reporting, and of course, a serious commitment for all concerned. So let me spend the rest of my time here today delving into some of the details of the Lumina-1 anti-activin A clinical trial. So exactly what is being studied here? We're studying new bone formation. We're studying flare-ups. We're studying the safety and tolerability of the activin A antibody. And we're studying function and overall well-being. So these are some of the key inclusion-exclusion criteria. Up to 40 adult men and women with FOP who have the classic R206H mutation. Again, that represents about 97% of patients with FOP. Subjects may have, but does not require, a flare-up at study entry. However, he or she must have had symptoms of FOP activity during the past year, and that's broadly interpreted. And finally, someone must be able to undergo all study procedures. In addition, that potential subject may not be concurrently involved in another study which involves imaging, blood tests, or experimental medication. So this is the slide I'd like to spend the most time going over, since this is really the study design. And there are basically three phases. There's a screening phase, there's a treatment phase, and then there's a follow-up phase. And the medication, which is the anti-activin A antibody, is given intravenously on a monthly basis. During the first period of the treatment phase, there's a randomization to either a placebo group or to the anti-activin A antibody. In the second treatment period, this is an open label period where everybody gets REGN 2477. So let me take you through the cartoon here. So once someone is interested in participating in this clinical trial, obviously we'll go through the inclusion-exclusion criteria. But once we bring that subject into our site, there will be an imaging study called a PET-CT. And that initial or baseline imaging study will help with randomization into the first treatment period. As I mentioned earlier, this particular imaging modality has the ability to detect not only existing heterotopic bone, but bone that is being newly formed. So it can detect activity of bone formation. And so that's one of the randomization criteria. that is being used in addition to things like someone's gender. So following the randomization, there'll be monthly infusions, either placebo or anti-activin A antibody during the first treatment period, which is about 6 months, and a total where someone will receive a total of 7 infusions. And then there'll be an open label period where everybody, again on a monthly basis, will receive the anti-activin A antibody. Following the second treatment period, there'll be a 24-week follow-up, which will include things like safety labs. The PET-CT scan will be performed 4 times during the course of the study, once at baseline to help with randomization, and then subsequently another 3 times. And just a word about this imaging modality. It would require someone to go into a scanner to lie flat for a variable length of time, probably on the order of 40 minutes or so, perhaps a little bit longer. And the scanning is preceded by an IV injection of a tracer that allows the activity of new bone formation to be detected. So what are these study procedures? There are visits to the study site, again, approximately once monthly, coinciding with the infusions. There's a physical examination that includes something that we call a CAGIS, which is an evaluation of joint function that really doesn't take much time, and it's performed by the site, one of the site investigators. There's the PET-CT imaging, which I just described. Of course, there's the medication, the investigational drug, and blood tests, which include safety and other blood tests. And then there are questionnaires regarding subject well-being and quality of life. So, not to state the obvious, this is a study of a new and unapproved medication. Its safety and tolerability profile is not yet fully understood. And the specifics of safety considerations will be reviewed with each subject in detail during the informed consent process. And the site investigator will be available to answer your questions about this. So for more detailed information, you can go to www.clinicaltrials.gov, and the title of the clinical trial is given on this page: a study to examine the safety, tolerability, and effects on abnormal bone formation of REGN2477 patients with FOP. So if you type in any of these as keywords, you would find this. And so just to wrap up this presentation, I want to thank the Regeneron Activin A research team, the R2477 study team, the Illumina-1 study team, and most importantly, I want to thank the IFLPA for allowing me to give you this overview on this exciting new investigational drug. Thank you.
Okay, so we will now start the Q&A portion of the webinar. You can, at the bottom of your screen, you'll see a button that says Q&A, and you are welcome to just click on that and submit your questions that way. So the first question that was submitted is, what are the differences of Regeneron's drug and Clementia's palovarotene? Not biologically, but clinically.
So I think it's hard to answer that question just in terms of clinical outcomes without talking a little bit about the biology. The two trials are at different stages in investigation, and so at this point it's sort of an unfair comparison because we don't have a lot of the early data that Clementia has. So what I can do is I can talk a little bit about the two studies in terms of who might be eligible, in terms of what are the requirements, and in terms of the type of drug that it is. And so, I will have to talk a little bit about the biology, that is how the drugs work, and from based on preclinical studies. So let's talk first of all about the requirements to participate. So in the Clementia MOVE trial, pediatric patients are eligible to participate in that study. In the Regeneron study, we're recruiting subjects aged 18 to 60 years old. So that's a big differentiator, if you will. So no pediatric patients in the Regeneron trial, but yes in the MOVE trial. In terms of what are the expectations, in both studies, you're expected to have site visits and safety labs, and a lot of the outcome measures are similar. The biggest difference is in the imaging modality. In the Clementia study, the primary outcomes are really based on a whole body CT, and so a CT scan can really measure the amount of bone, and so the primary outcome in the MOVE study will be based on how much bone, total body heterotopic bone forms. In the Regeneron study, we are also measuring the amount of bone, but because it also includes PET as part of the PET CT, we get activity of active areas as well as whole body heterotopic bone. The Regeneron study requires monthly visits because it is an infusion. In the Clementia study, the investigational drug is an oral drug, so that obviously does not require visiting the site to get the investigational drug. And in terms of side effects, we're learning about what those might be in the Regeneron study. We have preclinical data that suggests that there may be some side effects related to male genitalia, specifically the epididymis and perhaps sperm count, although that data was generated only in rats. That toxicology data was only in rats, not in non-human primates. In the Clementia palovarotene study, the side effects are limited mostly to the skin and mucous membranes. So things like rashes, some hair loss, some dryness of the mucous membranes, and dry eyes, things like that. Mucocutaneous side effects. So I think that's the way I would compare the two studies based on what we know today.
Okay, thank you. The next question is about whether or not if there's an FOP patient who's participating in another interventional trial, can he or she drop out of the study to join the Lumina study after a washout period? And if so, how long is the washout?
So the short answer is yes. There needs to be a period of 30 days or 5 half-lives, whichever is longer. And if, for example, someone was participating in the palovarotene trial, the longer of the two would be the 30 days.
Okay. The next question is about the study sites. Where will they be? And then if I can't get to one place, is there another location I could go to participate in the trial?
Sure. So as of today, there is one site in the US, here at the Mayo Clinic in Rochester, Minnesota. There is a site in Italy. There's a site in the Netherlands, and there's a site in the United Kingdom.
And the second part of that question is, if I can't get to one place, am I able to travel to another location to participate in the trial?
Sure. So I guess it depends where you are, but certainly if you were in the UK, there are 3 sites in the UK. As of today, there's only one site in the US. So the options, if you lived in the US, for example, if you couldn't or didn't want to travel to Minnesota, you would have to wait until another site in the US was open.
Okay. The next question is about the scans. The question is about whether or not if you need to be able to lie completely flat in order to be a part of the study.
Yeah, so you don't need to be able to lie completely flat, but you do need to be able to fit within the bore of the scanner. And so every scanner has an internal dimension. For example, the scanner we have here at Mayo Clinic is 70 centimeters. So you have to be able to lie flat for, again, at our site, you have to, and it varies a little bit with how tall you are because of how the scan works. But if you're of average height, we could probably do the scan in about 40 minutes. So you'd have to be able to lie flat for 40 minutes. And we help you, we help to make you comfortable by positioning and using cushions and pillows and things like that. But you don't have to be able to lie completely flat. You need to be able to fit within the scanner.
Okay, great. The next question is about the REGEN, the Regen 2477, the medicine itself. Does the medicine actually stop the flare from starting? Is it stopping the symptoms or just the bone from forming?
Right. So it's not perfectly clear from the preclinical data where exactly activin A works on the different stages of lesion formation in FOP. But it's probably very early because from the histological data that I've seen that has been published, you know, the activin A antibody is very effective at preventing early lesions from occurring. Having said that, you know, a drug like palovarotene, we know that works on the cartilage stage in FOP lesion formation. So translating that, we think that the activin A antibody could conceivably prevent the flare-up from happening. And if it didn't do that, it would likely work very early after that. We don't know, but because of the PET CT scanning, I think we'll get a very good idea about where the activin A antibody will work in preventing heterotopic bone.
Okay. And then the next question is about the study parameters, whether that's available worldwide or only in the US. And I think you've addressed that question in talking about the clinical study sites. So the next question, then we'll move on to, is it known at this time if the antibody will interfere with healthy tissue organs?
Yes, at this time, we have no safety signals that would indicate that that's the case. We wouldn't give the medication to pregnant women because there is there would be an effect on the fetus, but we're not aware, outside of the potential, as I mentioned, in the male genitalia. We're unaware of any other tissues that might be a target.
The next question is back to a question about the scan. Is it safe to say if we can fit into the CT scan machine at our local hospital, then we can fit in the machine, the PET scan machine?
Well, you know, we go one better than that. Before we would even bring you to the site, we would ask you to do two things. We would ask you to, you know, lie on your favorite couch or in your bed and send a picture of what that looks like. And then if let's say when you're lying flat, if you know, your knee can't be perfectly outstretched, just you know, with the tape measure, just measure from, you know, the surface of the bed or couch to the top of your knee. We would ask you to do a similar thing if, for example, it wasn't your legs, you know, maybe there was an arm that was locked in a position that made it outstretched from the, you know, from preventing you from keeping it close to your body. We would ask you to take a measurement and tell us the, you know, what your wingspan would be, and then we could easily tell you whether or not you'll fit in the scanner. So we don't like to guess because, you know, I don't know what your local CT scanner looks like. I do know what our PET scanner, our PET CT scanner looks like here. So that's usually the approach we take.
Okay, the next question is about the, back to a question about the medicine. Is it a repurposed drug or a brand new drug?
So it's not available. You can't write a prescription to get it. So it's not an existing drug. Regeneron is exploring the use of the same drug for other conditions, but this is not a repurposed drug in the sense that it's currently available for something else, for another indication. But it was on hand, as I mentioned when I was talking about the early preclinical studies, it was on hand and readily available to do the studies in the FOP mouse model.
Okay. The next question is also about the medicine. You indicate that it needs to be injected intravenously. Does that mean I will need to attend a site to receive the medicine? Are there any discussions about doing it home to save a trip?
Yes, so for this clinical trial, you would be required to come to the site to receive the infusion. At some point in the future, that might change, but for now, and because we're learning about the drug, this is the first clinical trial using the anti-activin A antibody, you are required to come to the site.
The next question is about the impact of the drug. Does it impact in any way normal bone growth?
So again, relying on our preclinical studies, I'm not aware of any effects on the normal skeleton.
Okay. The next question is about whether or not there are any chances of new sites in different countries. Will you be adding new sites?
Yeah, I think the short answer is yes. I can't speak to where those sites will be or how close they are to opening. Scott Mellis might want to address that, but to my knowledge, this is an ongoing process and there will be additional sites moving forward.
Yes, hello everyone. This is Dr. Scott Mellis. Yes, I'd like to confirm that statement. Regeneron is in discussion with multiple sites and getting close to opening other sites for enrollment, but we'll provide the details on that really when they're ready for prime time.
Next question is about the infusion and how long does it take?
So the actual infusion we usually do over 60 minutes. However, we have an observation period after the infusion is complete. So even though the transfusion might be, the infusion might be 60 minutes, for the first 2 to 3 infusions, we usually like to observe someone up to 4 hours after the infusion to monitor closely for any possible reactions. After that, we will not have such an intense monitoring. But the infusion itself, in answer to the question, the infusion itself is an hour.
You mentioned the CAT scans and checkups. Will that happen every month or every 6 months?
There are 4 PET CT scans over the course of the entire study. 1 at baseline, and then 1 at the end of the study, and then two in between. So a total of 4.
Okay. And then the next question is whether or not there is a timeline for a pediatric study.
Yeah, I'm unable to answer that question at this time. I'm not sure Dr. Mellis would be able to either, but you know, I think if this first trial gives an efficacy signal, I think that would be something that that Regeneron should very seriously consider.
Yes, hello, this is Dr. Mellis again. Surely pediatric studies are very important, and we hope to move as rapidly as prudent based upon the emerging safety and efficacy data that comes out of this study.
Okay, other questions? These have been great.
Yes, hello, this is Dr. Mellis again. Dr. Pignolo, would you comment a little bit more about the inclusion criteria with respect to whether or not a person can be experiencing a flare-up or a recent flare-up in the Illumina-1 clinical trial?
Sure. So again, the inclusion criteria will recruit up to 40 subjects, ages 18 to 60. There needs to be some FOP disease activity within in the previous 12 months. And of course, you'll need to be able to sign an informed consent. And of course, you'll need to have the ability to comply with the study's procedures and activities and come to the sites. But in terms of the the criteria, it does not need. We're not requiring someone to have had a flare-up necessarily in the previous 12 months. If you've have had a flare-up in the previous 12 months, that certainly counts as FOP disease activity, but it could be disease progression as defined by diminished activity about one or more joints. It could be defined as decrease in other functional parameters related to your FOP. So the key here is it doesn't have to be a flare-up per se, and that makes a lot of sense to me, again, because as I said earlier, almost 50% of patients with FOP don't progress, or do progress in the absence of flare-up. So this is a more liberalized inclusion criteria to capture all of those individuals.
Okay, the next question is about the dosage of the drug. Is it different based on weight?
No, the dose of the drug is fixed, and for this particular trial, the dose of the drug is fixed.
Okay, and then related to that, is there a different dosage if you happen to have a flare while you're in the trial?
No. There's no flare-up dosing.
Okay, perfect. The next question is about when the study starts.
So the study has already started, and there are 3 sites in Europe and 1 in the US that have enrolled patients.
We've had a few similar questions asked, so we won't repeat that. The next question was just about getting kids into the study, but at this point, this is not a pediatric study.
Correct.
Any other questions? We'll give you another minute in case you have any other questions. Okay, I think that is it for questions. So thank you so much to Scott, to Dr. Pignolo for being with us today. Adam, do you want to close us out?
Sure, yeah. Just would also like to thank Drs. Pignolo and Mellis for this very informative webinar. I think by the number and nature of the questions, it appears that there's a lot of interest in this study, the Illumina-1 trial and Regen 2477. If you have additional questions, feel free to email myself, Adam.Sherman@ifopa.org, or Dr. Pignolo, and we'd be happy to get a response back to you. So thanks to everyone for participating in today's webinar and joining in. Again, we'll have a recording of this webinar, which will be made available soon, should you like to go back for additional information. So thank you and enjoy the rest of your day.
Adam, there's one more quick question, if we have time to ask it.
Sure.
So the last question is, if and when children can take part, can they be sedated via other means during the IV or blood draws?
So that's a good question. Of course, this is a challenge. Part of the answer depends on what will be the lower age limit for participation in such a trial, which we don't know, but I would imagine that children of a certain age and maturity would not need sedation. There are some other ways that not using sedation, where children get studies that involve imaging and lying still for a period of time. They include devices where they can watch videos while having an imaging study. And it also depends on the type of imaging. If the imaging of a future trial involving pediatric patients involves just a whole body CT, for example, we can get those types of studies done relatively more quickly than something more elaborate like a PET CT. So just because we're doing a PET CT in this study doesn't mean that in a future study involving pediatric patients that the imaging modality will be the same. So there are a lot of variables to consider in the pediatric population. One is, what is the imaging modality and how long will it take? How old will the youngest age of subjects be in terms of inclusion? You know, what can we do besides sedation to keep a younger subject still? So I think there are lots of variables here to be considered, and you know, sedation, in my opinion, would be the last resort.
Okay, well, that will be the last word today. The video will be posted, of the webinar will be posted on the website in the coming days. And you're welcome to listen again or to share it with others in the community that you know weren't able to join us. Thank you so much for your time today. And thank you to Regeneron and to Dr. Pignolo for the work that you're doing.
Thank you.